Thanks. So it's great to be here. I was actually born in India many years ago, and I've lived outside of India most of my life, but visited regularly. And on this visit, it warms my heart to see ICTS doing so well, to provide such a wonderful international venue for interactions and bringing science together. Just walking around the campus, it seems like a temple built in utmost devotion to the theoretical sciences, and a non-denominational one at that, spanning so many different disciplines. So it's absolutely wonderful. Congratulations to ICTS, and thanks for the invitation. So I'm going to talk about a topic in theoretical neuroscience, but since it may not be so familiar to everyone here, I'll begin my talk with kind of a tour, a biased tour of the field. So why do I think theoretical neuroscience is such an exciting field? It's mainly powered at the moment by many experimental revolutions in our ability to probe the dynamics and structure of the brain across multiple scales of organization, all the way from molecules to overt behavior. And so we need to extract a conceptual understanding from all of this multi-scale data of how the brain works. So it's useful actually to turn to the physical and mathematical sciences that have themselves developed powerful tools to deal with complexity in their own domains. But also, neural systems aren't simply just a tangled web of complexity that exists for their own sake. They've been sculpted by hundreds of millions of years of evolution to solve important computational problems. So it's also very interesting to turn to the computational sciences for interesting metaphors or ideas about what kinds of computations the brain might be doing. Now, of course, none of these sciences are adequate in and of themselves, and so in confronting the complexity of the nature of neurobiological systems, neuroscience also provides a challenge back to all of these fields. So it's a very exciting subject that is extremely interdisciplinary, and we actually work at the intersection of all of these subjects as we try to understand the brain. There's also very nice, just to give you a tour of the types of things that we've been working on. So for example, we've been trying to understand how the retina processes natural scenes, the scenes that they've been evolved to process. Interestingly, the retina is actually in and of itself a deep neural network where photons control the activity of photoreceptors. It propagates through a hidden layer and generates spikes in ganglion cells that are sent to your optic nerve. It's very difficult to experimentally record from the interior of the retina, so we've been using techniques from deep learning and data where we look at the responses of the retina to natural scenes. We develop a deep neural network that computationally reconstructs the interior, and the interior of our neural network behaves in accordance with how the interior of the actual retina behaves based on the difficult measurements that you can sometimes do. We've also been looking at how the fly detects motion, right? Flies are incredibly difficult to swat because they're premier motion detectors in the animal kingdom. And people can actually record from the entire visual system of the fly using calcium imaging. And so using systems identification methods, we can try to identify the algorithm that the fly uses for calcium imaging. And interestingly, the last common ancestor of the fly and the human lived about 800 million years ago. And that common ancestor didn't have an eye. So that means the fly eye and the human eye evolved completely independently of each other. Yet, when we tried to identify the algorithm used for motion detection in the fly, we found that it was exactly the algorithm for motion detection used in the human, despite prior belief that it might not be. So that's sort of a fun project. Rajesh talked about hexagons. It turns out hexagons are in the brain of mice and bats and even humans. So as a mouse is traversing a square container as it's moving around, there are cells in the mouse's brain that fire if and only if the mouse is at the vertices of a hexagonal grid. That's quite remarkable. It was the subject of a Nobel Prize that was won recently in physiology. So this is a system, this is a brain region, the entorhinal cortex, that tells you where you are, right? So we've been working on theories and experiments of how this system is working. And how do you know where you are? You get information about how you moved in the past and landmarks that you see visually or through other sensory mechanisms. And you have to fuse these two pieces of information together to determine an internal estimate of where you are. So we've been working on experimentally testable theories of how neural networks can combine this information fusion between landmarks and idiothetic self-motion cues. And they make kind of testable predictions in neural data. We've also been working on how monkeys reach. I'll talk about this in much more detail in a bit. And actually, interestingly enough, the augmentation of learning is something that people are actually working on. People are looking at mutant mice that can sometimes learn faster, so i.e. they're smarter, but sometimes they don't do as well. Okay? So in a recent collaboration between 3 different labs, they actually developed mutant mice where they could, the same mutant mouse could either learn faster or slower depending on prior experience. And so basically, how well you learn is based on a mix of both nature and nurture. And we developed theories for plasticity, history-dependent plasticity mechanisms that could integrate both nature and nurture to determine learning speed. And they made testable predictions and data that were verified. So these are the kinds of things that they're driven by experimental revolutions we can actually do with the help of some theory. Each of these could be sort of a 1-hour talk, but there's another sort of very interesting interaction between neuroscience and machine learning. I'd like to argue that there's a motivation for a new type of alliance between theoretical neuroscience and theoretical machine learning, which is given by the following line of argument that we kind of laid out in this current opinion piece, which is we're all doing neuroscience because we want to understand how the brain works, but it's not even clear what that statement means. What does it mean to understand the brain or a neural circuit? A proximal version of that question is we'd like to understand how the connectivity and dynamics of a neural circuit give rise to behavior. And of course, learning is extremely important. We'd like to understand how neural activity and synaptic learning rules conspire to self-organize useful connectivity that subserves behavior. Now, there are many, many international brain initiatives that are promising large amounts of data that will speak to these issues. But actually, the field of machine learning has generated a plethora of learned neural networks that accomplish interesting functions, functions that we know of no other way of accomplishing in any artificial system at the moment. Okay? What's interesting is that we know everything about them. We know their connectivity, we know their dynamics, we know their learning rule, we know the entire developmental experience of these networks through their exposure to training stimuli, yet we don't have a meaningful understanding of how they learn and work, nor do we even have a benchmark for what that understanding would even look like. So I don't think for a second that if we understand how these artificial networks work, we understand how the brain works, but they form an interesting playground or warm-up problem for trying to confront the question of what it means to understand how a distributed nonlinear system computes. And so we've actually been, over the past few years, we've actually been working on a whole bunch of projects going from physics to neural networks where we use ideas from, similar to the way that Jennifer Chase just talked about it, where we use ideas from physics to try to come up with an understanding of how these networks work. And oftentimes, these ideas from physics lead to better learning algorithms. I just talked about this a couple of weeks ago at ICTS at a statistical physics and workshop. So, using ideas from non-equilibrium thermodynamics to create generative models, using ideas from random landscape theory to get better learning algorithms for neural networks, and so on and so forth. All the papers are on the website, and you can see the talk if you want. But I'm just saying there's a beautiful sort of potential for a symbiosis between these fields. And as ICTS grows and expands, I think this would be an extremely exciting growth area. What I do want to talk about today is underpinned by this field called high-dimensional statistics, which is motivated by a revolution in the way that we now collect and analyze data that really makes us have to think in very high dimensions. So what is this revolution? So when we used to do experiments, or back in the day when we would carefully decide ahead of time what were the important variables to measure. So for example, if we wanted to understand how a single neuron worked, we knew that the membrane voltage and the current across the cell membrane were two extremely important variables. So the dimensionality of the data was roughly 2. That the dimensionality of data is the number of simultaneously measured variables you take. And we measured, or Hodgkin and Huxley measured, those variables in as many, many situations as possible. So the number of data points was quite large. So this measurement density, which is the ratio of the number of data points to the number of dimensions, was very, very large. So this is the classical statistical limit, where the dimensionality is order 1, the amount of data is going to infinity, so the measurement density is going to infinity. And in this situation, it's extremely easy to do pattern recognition, for example, finding 2 clusters, finding a regression line, and so forth. But now what we do often in many different fields is we take data first and we ask questions later. For example, in neuroscience, we might stick a multi-electrode array into the brain and record from hundreds to thousands of neurons, the firing rates of these hundreds of thousands of neurons. So the dimensionality of our data is quite large, but we may not be able to take that many measurements before the animal gets tired. So the number of trials that we get could also be quite large, but the ratio between the dimensionality of our data and the number of trials, i.e. the measurement density, is now order 1. The simplest way you could visualize this is like 3 points in a 3-dimensional space, and you might wonder how we can ever deduce anything from such high-dimensional data sets. So this is a very beautiful field, and there's actually a beautiful connection between theoretical methods of understanding algorithms in high-dimensional statistics, and the statistical physics of quench disorder. And we've been working on some of these things. But I wanted to talk about just one work now that shows how simple ideas from high-dimensional statistics can have implications for the design of experiments in neuroscience and for conceptual understanding in neuroscience. So, and by the way, for physicists who are interested in sort of learning about this subject, we actually wrote a sort of a 70-page review that covered in a unified way using replica theory a whole bunch of topics in both neural network learning and high-dimensional statistics, random matrix theory, compressed sensing, and so on. So this might be a fun reading for some students in the audience. Okay, so what I wanted to talk about is what I think is a major conceptual elephant that resides within almost all works in systems neurobiology, which is the following question, which is basically the question of what the heck is it that we're doing in this field, right? How is it that we can record order 100 to order 1,000 neurons in regions deep within the brain, far from the sensory or motor periphery, and yet obtain scientifically interpretable results that relate neural activity to behavior and cognition? For example, we can create brain-machine interfaces that allow paralyzed patients to control a robotic arm by only recording 100 neurons from the motor cortex of that patient's brain. This is absolutely remarkable, considering that these brain regions contain order 10 to the 6th and 10 to the 9th neurons, about 5 orders of magnitude more than we can record. So the question is, how has systems neuroscience been as successful as it has been in such an undersampled measurement regime? Or is it that we're completely misleading ourselves and our conceptual view of how the brain operates may completely change as we record many, many more neurons? Okay. Lest you think that there's a quick experimental solution to this problem, while it is the case that we've been experiencing an exponential Moore's law type kind of growth in the number of neurons we've been recording, the doubling time is about 7.4 years, right? So in order to get about 5 orders of magnitude more neurons, we'd all have to show up here at ICTS 130. ICTS will probably be around, but we may not be, unfortunately. So what we need is a theory of neural data analysis that tells us how and when statistical analyses applied to a small subset of neurons reflect the collective dynamics of the much larger unobserved circuit that they're embedded in. Okay. So I'll give you such a theory, and we're going to apply it to a certain data set. What is that data set? It's a monkey just performing reaches. So the monkey is holding a target cue, and it performs a ballistic reach to a certain point. And during these reaches, they're recording using a multi-electrode array that enables them to get about the firing rates of about 100 cells. Okay? What do these individual cells look like? So these are the firing rates as a function of time for one cell, a second cell, a third cell, for multiple reaches in different directions at different lengths at 2 different speeds. And as you can see, there's a lot of complexity and heterogeneity in this data set. It's very difficult to understand this data set at the level of individual neurons. So how are such data sets analyzed? What people do is they do dimensionality reduction, for example, principal components analysis. There, the basic idea is that every pattern across neurons that occurs at any given time is a time-dependent linear combination of a static set of basis patterns, the principal components. And oftentimes what happens is all the neural activity patterns that occur throughout the entire experiment could be explained using a linear combination of a very small number of basis elements, basis patterns. So the number of basis patterns required to explain a decent fraction of variance in the data is often quite small. And then you can plot the data in terms of the amplitudes of the patterns, and you can get a neural state space dynamics. And this neural state space dynamics actually often gives you a remarkably insightful dynamic portrait of what's going on in the circuit while it's carrying out the computation that it's supposed to do. So this is how dimensionality reduction is often used in the brain, in neuroscience, to visualize neural circuit dynamics. These are examples of different neural state space dynamics, and they teach us actually quite a lot. For example, when the locust is smelling a new odor, its activity in its antennal lobe tells you that the transient dynamics of the circuit contains a lot of information about odor identity long before it settles into the fixed points. Also, when you record from the motor cortex for the prefrontal cortex of a monkey doing a complicated decision-making task, it turns out the curvature of these trajectories gives you a clue into the algorithm that the monkey is using to perform complex decisions. If you record from the entire brain of the zebrafish, where you can get about 80,000 neurons using calcium imaging, remarkably, you can fit the entire dynamics into 3 dimensions to a good approximation. So anyways, this is what's done a lot in the field, but this raises some fundamental conceptual questions. how the heck can we trust these dynamic portraits of circuit computation despite recording so few neurons? How would the shape of these portraits change if we recorded more neurons? Why is their dimensionality so low? What is the origin of this intrinsic simplicity in neural dynamics? Would it increase if we recorded more neurons? And this is a question that really extends beyond the writ of neuroscience alone. It's really stated abstractly. It's what if anything, can we learn about large dynamical networks at such an overwhelming level of undersampling? Okay. And actually, what we'll find is we'll find a predictive theory of experimental design that can tell us how many more neurons we should record as we move to more and more complex tasks. So let me just tell you the take-home messages of the theory. So basically, we define a new quantity, which we call neural task complexity, that takes into account two pieces of information: the complexity of the task that the animal is performing and the smoothness of neural dynamics. And then with this mathematical definition, we prove a theorem that the dimensionality of neural activity is actually upper bounded by this neural task complexity measure. And then we go back to the motor cortical data, and we actually show that despite being so low dimensional, the motor cortical data is actually, relative to the number of recorded neurons, the motor cortical data is actually as high dimensional as possible given the simplicity of the task. And this actually has a cautionary tale, which is in future experiments, if you simply record more neurons without changing the task, you won't at least get richer data sets, at least as measured by the dimensionality of the data set. Now, on the issue of the shape of these trajectories, we derive conditions for accurate recovery of these dynamic portraits. And we use the theory of random projections to show that the number of neurons required need only grow logarithmically with this neural task complexity. So this is fantastic news. This is saying that we can trust the wide body of work in neuroscience that looks at state space dynamics in the brain. And it shows that we don't need as much experimental resources as we might think we would to get accurate recovery of these portraits as we move to more complex tasks. So what is this neural task complexity? So imagine, just for intuition, imagine that a monkey is reaching to multiple directions, okay? It has a manifold of behavioral task parameters that corresponds to, in this particular case, a cylinder, which is time by angle, okay? And the average neural activity in the brain, averaged over many reaches, is fundamentally an embedding of the cylinder into the high-dimensional space of neural activity patterns, where this dimensionality of the ambient space is the total number of neurons in the circuit, okay? Now, just intuitively, skipping all the details, if this manifold doesn't have that large of a volume, and if it's not that curved, then it can't explore that many linear dimensions. So the neural task complexity is literally the volume of the manifold in parameter space divided by kind of an autocorrelation cell over which it doesn't bend very much. Okay, and it's quite intuitive that this might upper bound the dimensionality, and we can make that rigorous. Okay? So if we go back to the actual neural data, it turns out that when a monkey is reaching, there's 100 million neurons in motor cortex, there's 650 skeletal muscles. The dimensionality of the brain's dynamics is only 7 if you look at all 8 reaches. So that's quite small relative to the total number of neurons, which is about 100. Okay, but if we compare all of this to the neural task complexity, we find that actually, because the reach is so simple, the highest the dimensionality could be is about 10 for all 8 reaches, and that the dimensionality that actually reaches is 7. Okay? This actually makes some predictions that would be non-intuitive to neuroscientists, which is if you vary the task complexity by varying the duration of the reach, then if the complexity of neural dynamics hugs the complexity of behavior, then the dimensionality should be proportional to the duration of the reach. Okay? Second, if you just throw away neurons in the data set, then the dimensionality should remain unchanged, because the number of recorded neurons is not the bottleneck for determining the dimensionality of brain dynamics. It's the simplicity of the task that is the bottleneck. And both of these both of these predictions get verified. Here we see that the actual dimensionality of neural dynamics in motor cortex does hug the complexity of the task as measured by the duration of the reach. And also, if you drop 60% of the neurons in your experiment, the dimensionality remains unchanged. All right, now what about the shape of these portraits, right? Now, how is it that we might get the shape of these portraits correct, even if we're recording 100 neurons out of 10 to the 6th? So what's the simplest situation in which you can visualize why you might get this correct? Imagine a circuit that has 3 neurons alone, right? And imagine, for whatever reason, the neural dynamics is low dimensional and not that curved, okay? And furthermore, imagine that this neural manifold of states visited by the brain during the task is not axis aligned with respect to the single neurons. Okay? Now, you don't get to measure all 3 neurons. You only get to measure 2 neurons, let's say. So you only get to see a shadow of the neural manifold onto the subset of recorded neurons. And here it's intuitively clear that no matter which shadow you see, the geometry of the shadow will be similar to the geometry of the full manifold that you would have had access to if you had access to all 10 to the 6 neurons. Okay? Of course, the picture may not be this rosy, right? The manifold could be axis aligned in the sense that neuron 2 is doing all the work. And then whatever subset of neurons you record better include neuron 2, otherwise you'll be in big trouble. Okay? So this is the intuitive picture, but can we make this much more quantitative? So here's the basic idea. Let's assume that this is what's actually going on in the brain, and then pursue the consequences of that and try to extract testable predictions. So let's assume that if the neural manifold is randomly oriented with respect to the single neuron axes, if we assume that, then an experiment that everybody is doing right now, which is measuring a random subset of neurons, becomes equivalent to an experiment that nobody is doing right now, which is measuring m random linear combinations of all neurons in the circuit. Why? Because geometrically, this experiment corresponds to a projection of the neural manifold onto a randomly oriented subspace, while this corresponds to a projection onto a coordinate subspace. If the neural manifold is randomly oriented to begin with, the orientation of the subspace of the projection doesn't matter. Okay? If that's the case, then we can exploit ideas from random projection theory to discover scaling laws for experimental design in neuroscience. So let me just review that theory. So what happens often is that signals of interest that we care about often live in a very high-dimensional ambient space, but they're constrained to a much lower dimensional manifold. And so if you project that manifold onto a random subspace, the geometry of the projection doesn't incur that much distortion. So imagine, for example, that you have 2 points in the high-dimensional space, and you project them down to the low-dimensional space. Okay? Let's say this space is k-dimensional, and you're projecting from n down to m-dimensional space. The distortion is the fractional error in the distance measured in the low-dimensional space versus the high-dimensional space. And what we care about is the worst-case distortion over all pairs of points. Okay? There's very beautiful theory that shows that with high probability over the choice of the random projection, as long as the number of neurons or the dimensionality of the subspace is bigger than order 1 over epsilon squared, which is the desired distortion level that you want, times the intrinsic dimensionality of the manifold, times the logarithm of the curvature times the volume, then you will indeed achieve the desired distortion, okay, with high probability over the random choice of projection. Okay, so this is actually a quite striking result. What it's saying is that you don't need to, it's actually largely independent of the ambient dimensionality, which is the total number of neurons in the brain, or the brain circuit in our application. It only depends on the intrinsic dimensionality of the manifold and its curvature and volume. So this is potentially good news. And when you translate this into the language of neuroscience that we've been using, this makes a striking prediction. Okay? It says that the number of neurons that you measure scales as 1 over the distortion squared, right? This is the distortion between the state space dynamics you get using the number of neurons you actually record versus what you would get if you recorded all the neurons, times the logarithm of this task complexity. Okay? So this actually makes a testable prediction in the brain. This is saying that isocontours of constant distortion are straight lines in a plane formed where the vertical axis is the number of neurons that you record, and the horizontal axis, in the case of reaches, is the logarithm of the time of the reach. Okay, so ideally, we'd like to record all the neurons in the brain, or in the motor cortex, in the most complicated task, but we have a finite data set where we have 109 neurons in a fixed duration task. So what we can do is we can subsample neurons, compute the state space dynamics, and compare the accuracy in the subsample state space dynamics compared to what you would get if you had access to all the neurons that we have access to, namely 109. And then we can do it for reaches of different durations. And the isocontours of constant distortion should be straight lines in this plane. Okay? We could be more generous and allow ourselves more neurons to get a lower distortion, but we'd have a similar isocontour. Okay? So we can actually test this in real neural data. Now, this isn't an empty test of a mathematical theory that already has a theorem guaranteeing it. What we're really testing are the assumptions underlying the preconditions for that theorem, which is namely the neural manifold of states explored by the brain while the monkey is doing this task is indeed randomly enough oriented with respect to the single neuron axes such that random projection theory constitutes a good theoretical model for the active electrophysiology of recording a random subset of neurons. And so strikingly, this is actually verified in real neural data. So this is the logarithm of the reach duration, or more generally neural task complexity, or in logarithmic axes, and this is the number of neurons recorded in linear axes, and as you can see, the isocontours of constant distortion are straight lines. So just to give you the take-home messages, we've developed a theory that quantitatively explains one of the fundamental descriptors of the complexity of brain dynamics, at least its dimensionality, and we've compared, and this kind of tells you that if you want to understand if the dimensionality of neural dynamics is due to intrinsic constraints in the neural circuit, you have to show that it's much lower than the upper bound provided by the simplicity of the task. And actually, this reveals quite optimistic messages about us being able to accurately recover dynamic portraits of circuit computation in the brain, even in more complicated tasks, without recording that many more neurons. I have some philosophical messages, which I will just leave up here for now. Thanks.
Okay, questions, comments?
So I have a bit of a naive question. So you define neural task complexity. For some task, which we don't know how the mechanisms in the brain, how do you know whether it's high or low relatively? Because in the brain, you might have a conjecture like it's in this part of the brain, but it could be a distributed mechanism also.
Right. So the only aspect of neural dynamics that entered in the definition of neural task complexity was roughly the smoothness of the neural dynamics across task parameters. And that's
smoothness?
So smoothness, I mean, so if you look at these, it's easiest to understand in these single neuron. Where did it go? Yeah. So these neural traces as a function of time or task parameters have a certain autocorrelation length, i.e. you have to wait a certain amount of time before the pattern of neural activity changes appreciably. That's an autocorrelation length in time. Similarly, across different angles, there's a spatial autocorrelation length. These have remained remarkably invariant across animals, across tasks, and so on. So it's just that number that you need to know, and you can often estimate it before you do the experiment. And if you want to be conservative, you can underestimate it, give an overestimate of the neural task complexity, and an overestimate of the number of neurons you need to measure. So it relies on very little information about the dynamics in the brain. Of course, no theory can assume nothing about the dynamics in the brain. So this is, I think, actually the minimal thing that you need to assume to get these kinds of results.
I'll take a question on this side and then on the other side.
So Surya, very nice work. Could you speculate on what happens when the brain is not homogeneous in terms of its neurons? And we know that the brain has many different kinds of neurons, or even the cerebral cortex or the motor cortex.
Exactly.
How does that map onto the number of neurons that we need and the dimensionality of a given fixed task?
Yes. So we've been looking at brain regions like the motor cortex where there's a lot of heterogeneity, statistical heterogeneity, where these are examples of sampled neurons, right? So even if there were some kind of topographic organization where neurons in one part of the brain region behaved a certain way and neurons in the other part of the brain region behaved a certain way, and your electrode covered both regions, these autocorrelation lengths are an emergent property of both regions together. So the theory would still go through in that case. But what you're alluding to is that these state-space dynamics may not be a complete theory of what we want to understand. And you will talk about much more detailed aspects of neural dynamics that goes beyond the state-space dynamics. But if you have heterogeneous or spatial topography, then what I would do is I would do the dimensionality reduction separately in both regions. Or that may not be the right way to think about it, right? People oftentimes parcellate the brain into anatomically defined regions like the frontal eye field and the parietal cortex, which work together to control eye movements. But actually, these two regions are bidirectionally coupled. So it might not make any sense to think of these two heterogeneous regions as operating independently of each other, there might be some emergent circuit that combines both of them. So it's an open question, what is the right way to parcellate the brain? What is the most parsimonious way to parcellate the brain in order to understand any given behavior? There is no universal answer to that question. It will depend on the behavior in question. But there's multiple ways to sort of extend the theory to heterogeneous cases. But you know, this state space dynamic is only the beginning. I mean, this is what a lot of people do, but there's a lot more detailed information that you could ask for as well.
Question here? Any other questions? Okay, if not, let's thank the speaker.